Hi there. It's Wellington, the producer. I'm here talking to you instead of Jane, because Jane is stepping away from the bar. She has to leave to take on a new research opportunity. We are so gonna miss her. Me most of all. But as you know from this podcast and others, it takes a team to get this to you. And so we're gonna continue the podcast, and we're gonna continue to bring you the latest science stories. Some of the next episodes we're planning for season 4 are going to be about asthma and multiple sclerosis. So stay tuned for updates from the team. The best way to do that is to subscribe if you haven't already. That way, you'll be the first to know about the new season. I can't wait. Now, in the meantime, we have some episodes we recorded with Jane before she had to step away. We hope you enjoy them. And as always, thanks for listening.
Because of the way these mutations arise in the tumor, for each individual patient, their pattern of mutations is likely to be unique to them, almost like a fingerprint.
The connection to full tumor burden, I think, has to be established, but the idea is certainly there.
I'm Jane Grogan, and I'm a scientist, specifically an immunologist, so someone who studies how the immune system works. One key part of my job as a scientist is to communicate ideas with other scientists and also with people outside of the field. One of the cool things is this podcast allows me to do both. For the past 2 seasons, I've had the privilege to speak to some of the brightest minds in research, but I'm not done yet. This season, I'm going back into the bar to see what my colleagues are doing to research some of the most complex diseases and see what they're up to. So grab your favorite drink, get ready to unlock your science brain, and join us for 2 Scientists Walk Into a Bar. So how do you use your phone to track your health?
I use my phone to track heart rate, calorie burn, weight loss, and also number of steps I walked.
Heart rate, weight, sometimes nutrition, sleep.
Breathing, steps, and all that good stuff.
I have recently been using it a lot just to see what things excite or stress me out.
I like those answers, and a lot of those things that were mentioned are biometrics and biomarkers. Biomarkers, you may recall we did a podcast on this earlier, but they're really critical for us in science to understand how a disease progresses and how we can treat disease. We thought biomarkers deserved another shot. And here to help unpack what biomarkers mean now and for the future are both a scientist and a medical doctor. I'd like to welcome Mark Lee and David James.
Thank you.
Fantastic. Thanks for having us.
It's an absolute pleasure. The title of the show is Beyond Biomarkers. There's so much to unpack here. Mark, what does beyond biomarkers mean?
Well, we think of biomarkers of the past as being more unidimensional, right? They were single markers that parsed the population into maybe a small number of segments. What we realize now is that there's more explanatory variables for each individual patient that require a multidimensional approach.
And I think the key point is that, it's really the integration of the different layers.
But we're already generating biomarker data, right? We're taking blood tests in the context of oncology. There's PET imaging for tumor size. There's blood tests. There's MRIs, all kinds of things. Why are these not sufficient?
It's fundamentally a struggle against heterogeneity, right? And more heterogeneity than we've been able to measure before, right? And our instruments in the past have been more blunt, and so we've not been able to go beyond these kind of subsets of the population and really make progress towards individualizing care.
Hi, Jane.
Hey, Wellington.
For a non-scientist, can you explain heterogeneity?
It's really just about diversity. Not every tumor will be the same in one patient or across patients. Not everyone's blood samples will be the same. I think what Mark's referring to is that how do we monitor that and track that? If you think about the way tumours are measured in the clinic, you look at the size of the tumour, and I think there's some discussion around, does that size represent increased tumour growth or just that you've got really good cells in there that are eating up the tumour? And so it's kind of fairly gross. But when you think about detecting tumour DNA in the blood, that's a big jump. What is the process by which we can detect this tumour DNA? And the blood's a pretty complicated mix of stuff as well.
It is. I mean, the reality is that all of us have some amount of this free-floating nucleic acid DNA primarily in the blood. And so white blood cells, which are nucleated cells, right, they have DNA. At some level, they turn over and release their DNA into the blood, okay? And this has been known for a long time. But in patients with cancer, if you're able to sequence the DNA, that's this free DNA in the blood, sequence it broadly enough, covering enough of the genome, and sequence it deeply enough so that you're looking at as many genomes as you can recover from the blood, what you find is that a very small amount of that DNA, in most cases, is actually derived from the tumor. And the way you know that is because it harbors these classical mutations that you also find in the tumor tissue.
So this is a really remarkable things about tumours that we've learned in previous podcasts as well. But that as they mutate and they have these truncal mutations or driving mutations, that they become very unique to the tumour, which you don't pick up in healthy tissue, even if those cells are dying. But the frequency of that DNA is very low. I mean, I imagine this as kind of turning a telescope out to the skies and trying to pick one star from out there and say, that's the one, that's it.
Absolutely. But to your point, though, because of the way these mutations arise in the tumor, for each individual patient, their pattern of mutations is likely to be unique to them, almost like a fingerprint. And because we can see that fingerprint at a very low level, albeit, granted, but we can see it in the blood, is actually a direct measure of the amount of tumor.
So David, when can we measure that relative to what we would normally see in a PET imaging scan, for example?
The connection to full tumor burden, I think, has to be established, but the idea is certainly there. I mean, it's not. I think an important precedent to it is what was done with many years ago with HIV. Essentially, the way that docs now diagnose the progression of HIV is through assays that measure HIV genomes.
So viral sequences
Yeah,
that are very
right.
different from the host.
Right, right. So that's in some ways easier because there's lots of differences with viral genomes than there are with trying to measure something in the person's actual cells. But the concept's the same, right? And so hopefully we'll once we get there with the data, that the connection between the presence of tumor DNA in the blood will link to important outcomes like overall survival. But we're not there yet, but I think the technologies are there.
Wait a minute, Jane. We can really find evidence of tumors in the blood?
I know, it's really cool. We call this kind of liquid biopsies, right? Being able to take a blood sample and looking at the free DNA that's floating around. And when you think about it, as cells turn over or die, they're releasing their DNA. So you can actually detect that in the blood. What's really amazing is now that we know the tumor mutations, we can create these genetic fingerprints of the tumor and then use that to probe blood DNA as well. It could have really remarkable effects on the way we diagnose patients. So Mark, how is the field using this technology to really personalize treatment and care?
Well, one of the things David and I have been talking about recently is the use of this technology in patients who are getting curative therapy. Like they have early stage cancers and then they're getting surgery, sometimes radiation, maybe they get some systemic therapy afterwards. And what we know in some patients, after they're done with all of that therapy, and you were looking the blood, some percentage of those patients turn out to have circulating tumor DNA. And we know that because we know what their original tumor looked like, and that fingerprint of mutations is there in the blood. And what we'd like to be able to do is direct new therapies at those, towards those patients to give them another chance at cure.
So they have minimal disease still.
Well, we can't see anything on the scans,
Right.
but it's there in the blood. It's like a needle in the haystack, but we've got the tools to see that needle.
And how far is the field in linking the presence of that DNA with tumor resistance or novel mutations that are arising in the same patients?
It depends, I think, on the setting. There's conceptual data, some anecdotal data out there showing, a pretty clear and direct correlation between the levels of these mutant alleles in the blood and overall outcome in terms of time to relapse. But big studies that really establish the precedent are probably being run right now, but there's no data like that's substantial yet.
And of course, it'll be wonderful when we can truly correlate this with tumor burden.
Oh, absolutely. You know, and I think you can imagine how, well, I mean, just in that setting, the setting of patients who've been treated for cancer and thought to be cured, where there's nothing on the scan, but it turns out the circulating tumor DNA is actually there. If we could administer therapies and drop that level of ctDNA, circulating tumor DNA, down to nothing, perhaps that's the way we know that those therapies are working.
Maybe that's the definition of cure then.
Yes, and today we are administering so-called adjuvant therapy. So in patients who've been cured and we give additional chemotherapy, for example, we don't actually know if it's working.
Or it's needed.
Or if it's even
I
needed.
think that's a really key point too. Like what's needed as well. Like knowing this information and combining this with other biomarkers will allow the patients to get the actual medicines they need rather than everything that's on offer just in case it's going to work.
Like today, you know, we might treat 100 patients with a certain stage of colon cancer. They all had their surgery, and we give them all chemotherapy. But to your point, you know, as David points out, not all those patients actually need to have additional therapy. And boy, wouldn't it be great if we were going to give therapy, give it only to those who need it.
Yeah. And I think maybe Mark might disagree with this, but I think, speaking of the future, you might not, but as of today, it may not be that the ctDNA actually correlates with tumor burden. It just may be a different endpoint, a different way of measuring whether there's a tumor there. You know, the fact of whether it actually correlates with the overall tumor burden is interesting. What really matters is how well it drives clinical decision making and ultimately whether it correlates better with overall survival.
Well, that begs the question then, what else do you need to measure to make that endpoint more solid? And are there other biomarkers beyond ctDNA that are really important?
Yeah, no, absolutely. By the way, David, you know I always agree with you. I don't disagree with you. Come on. Yeah, you're very astute. Yeah, but Jane, I mean, to your point, I mean, I think there's an opportunity to measure other features of the patient, especially over time. You know, one thing we're very interested in is imaging, right? It's taking full advantage of the data that's present in these scans. And while we have traditional ways of looking at scans and quantifying the amount of tumor and the amount of change across time, we're realizing that machine-based approaches, where we're taking advantage of every pixel in every one of those images, might actually be more quantitative and more revealing.
So going actually back to original data rather than necessarily creating new information.
Correct.
And reproducibility too, right? I mean, I think that's one of the, definitely one of the great advantages of automation is that you can do things. You're not relying on, the same pathologists on the same day reading the same scan 3 times the same way, right? You can have a computer do it. It takes a split second, and they're doing it exactly the same way every time. I think there'd be huge advantages to that, even if you're not even scanning people. If you're just scanning slides, looking at someone's pupil, it's just you can measure things the same way.
Yeah.
Which is really hard to do in science.
But, my experience of biomarkers is a little simpler than that. You get a biopsy and you slice the tumor, and there's only so much tissue and so much you can do with that.
Yes.
And things often get reduced down to 1 or 2 analytes or 10 analytes. But to get to this kind of granularity, we need to go broader with both the tumor sample, the imaging, as you were talking about, Mark, and then the ctDNA, as you were talking about, David, and then other things as well. So what do we do with the tumors and the biopsies, and how can we be better at using that material for biomarkers?
I think one of the hard parts of what we do is that you don't know necessarily what's important. And so you don't, right? So you have to test 50 things, and 25 of them look good, and then you retest them, and then only 5 look good. So it's just this iterative process. We're trying to figure out, what's important in the context of a particular drug. Sometimes it's more obvious than others. Like if you have a very targeted inhibitor, you kind of know that you need to look at the target. But it's probably the simplest case. When you have more complex systemic therapies like immunotherapy, you don't necessarily know that. So, one of the big changes from I think a few years ago is that everything we do now is multiplexed, meaning we don't look at one thing at a time. We look, you do whole genome sequencing, whole transcriptome analysis. Where we used to do, 1 or 5 or 90 PCRs. Now we can look at everything together, right? And so that's dramatically improved our efficiency with samples. It arguably has complicated the situation because now there's so much to look at. You don't know, unless you have large numbers of patients, what's real and what's not. So in some ways it makes discovery more difficult, but it certainly broadens the landscape of things that are potentially possible.
It's certainly the challenge and the appeal of the reverse translation as well.
Yeah.
So if we want to look at what's underneath the iceberg and how big it is, what kind of data is the field starting to collect? Because it could go on ad infinitum, right? And what makes sense and what should we be collecting at risk so that years from now we can look back the same way we're looking back at PET imaging, for example, and reassessing that. Because one starts to think about there's tumor sampling, there's blood sampling, there's saliva sampling, there's bodily fluid sampling, and the list goes on.
Yeah. Well, we have to place some bets, right? And certainly the sample acquisition is something that you can't go backwards in time to collect. So sample banking, I think, is going to remain important in these large studies, and that's what people are doing, right? But then we do have to commit to certain molecular profiling platforms, data analysis platforms, imaging. Then we have to figure out what we're going to do with those samples. And at this point, we are looking at germline, whole genome sequencing on the germline. We're looking at whole exome sequencing on the tumor. And RNA-seq, we're looking at ctDNA. I mean, these are the leading platforms. They're the most mature platforms. And I think we believe there is true information content in each one of those that's complementary.
Jane, what is sample banking?
Oh, it's really just being able to take samples from patients on clinical trials, maybe blood, it may be their tumor, and collect it and either analyze it in real time or bank it so that we can ask questions of it later. How do we collate these data sets, right? We've got the imaging, the whole genome sequencing. We've even got T-cell receptor sequencing that's coming online as well. And there are a number of different data sets across the world, whether it's in consortiums or held within academic or commercial organizations.
Yeah, so I think there just has to be an acknowledgement that not everything's going to be important, but there has to be, you have to put the investment out there or invest now to sort of collect the information while you're getting the samples. Over time, the value will accrue, I think. So for example, the Cancer Genome Atlas, which was a project sponsored by NIH a decade ago or more, where the idea was we were going to sequence everybody's or a bunch of tumor types. And I think there was an expectation that by doing that, everything will solve all the cancers that are out there, and it just doesn't work that way, right?
It's like even sequencing the human genome would be done, right?
Yeah.
We'd have solved all disease.
We haven't solved it.
No, not at all.
How do genes really work, right? And what regulates them? We're still working that out.
It is complicated, but I think you just have to take that risk. It's like a lot of what we do. It's just there's a risk of the unknown. Doesn't mean you shouldn't do it.
Yeah. Well, I think the other thing that we've learned over time is that the scale of the experiments matters so much, right? Even something as large as the Cancer Genome Atlas, when you divide it across 30, it's more than 10,000 patients, but divided across 30 tumor types, for each tumor type, you actually didn't have that many patients, right? And now we know that each of those tumor types is actually not one disease,
right,
but probably dozens of diseases, and so then the numbers get really small.
Which becomes a statistical problem or
Exactly.
a statistical a problem in
Exactly.
the context of heterogeneous biology. right? We've
Exactly.
got-
It's very likely that we would have needed the cancer genome atlas should have been 10 times bigger, 100 times bigger, for us to really make these analyses meaningful in the subsets of disease we now know exist.
But the reality is, as we collect these larger data sets, we still need to analyze and interpret them along the way when they're still small. And this is one of the challenges for biomarkers and diagnostic and treatment right now, is that in a quote unquote perfect world, we'd have all that data already, and we don't. We still need to generate it.
Yeah.
Yeah.
This is the real challenge, right? I mean, we talk about this, the need for meaningful data at scale. That you.
I knew you were going to throw that in.
I couldn't resist. I couldn't resist. No, but
I can give you that from the lab, right? We can grow cells in a dish or, look at various preclinical
Yes.
models. But when it gets to
Humans,
patients, it's very
but
different.
it's but this is the data that we need to really draw firm conclusions about associations between certain profiles and what ought to happen in terms of therapy, right? I mean, you're not going to get that from any other place other than from human, from populations.
So, Mike, we've been talking about oncology, but this is applicable to all kinds of diseases. How are you thinking about, or how is the field thinking about this in neurobiological diseases or inflammatory diseases?
So it's absolutely parallel, right? I mean, and there's definitely analogs for what we've done in oncology, and admittedly, oncology has been ahead of the pack in terms of this kind of biomarker-driven personalization of care. Maybe my colleagues outside of oncology, would take umbrage with that, but I mean, the reality is that that stratifying patients into different subgroups is something which we're pursuing with very similar approaches, looking at the genomics, looking at imaging. If you think about neurologic disease, can MRIs, of the brain be used to look at an anatomic approach to segregating disease, right? Could serial MRIs show us a change in the appearance of the lesions? Could that stratify patients? And would there be therapeutic consequences? Especially as the number of therapies grows in a particular indication, could we say, hey, therapy A works better in this strata of patients determined by imaging? And would therapy B work better in this other strata? So we're just beginning to learn how to do that. But that's very much analogous to what happened in oncology, with the advent of genomics.
Yeah, I think there's more recent examples of this, certainly in eye disease, right?
Yeah, absolutely. There's some beautiful work by a number of groups now where machine learning approaches to look at retinal images, which ophthalmologists have been acquiring for years, that it turns out you can learn more than whether someone has disease or not. You can actually learn other things about that patient, including predicting their disease course, which most ophthalmologists would not have guessed before. Some very nice work showing that a patient's gender, their age, their blood pressure, hemoglobin A1C, these are cardiovascular risk factors, are evident in the retinal image if you have enough data to train those models. And what we're learning now is in a patient with retinal disease, like diabetic retinopathy, can we predict which patients in a population are likely to have vision loss over time and which ones are actually going to be unaffected? I mean, knowing that would have big implications on when to intervene. And we're finding that that information is hidden in those retinal images, and now we can uncover them with data at scale.
Which is remarkable that there's such a crossover implication to, cardiovascular diseases or other implications. Machine learning versus artificial intelligence, right? Are we speaking the same language here, or does artificial intelligence portend more values, other interpretations than just using machines to evaluate data?
Well, I think we think of these as being related concepts, right? And there's certainly the opportunity to automate features in data that humans would observe, right? Like you think about pathology, we can develop algorithms to recognize cancer cells and lymphocytes and stromal cells, et cetera. And then there's this category of deep learning where the machines are not told to recognize human features, but to look for features on their own, to learn the features on the basis of ground truth, which in the case of patient data is outcome. Is there something they see in this collection of ones and zeros, these pixels in an image, that put together portends a certain outcome for the patient?
Or predicts it.
Or predicts it. And that then, it's a bit mysterious how that happens, right? We're not always able to figure out exactly how these models are making those predictions. But there's a lot of very exciting work to say, can we back out from those models the features that are most important? And could we learn new features, new elements of biology that wouldn't have been evident from the feature-based approach, right? Where we're telling the machines, here are the things we know, and assemble those things into a story about, these are poor prognosis patients and those are good prognosis patients. In this case, we're saying, we're telling you up front, here are the good prognosis and bad prognosis patients. Tell us what's hidden in these images that would explain that. And I think they're complementary approaches and very exciting.
So David, when you think about using these kind of data sets and fitting that information into this machine learning and algorithm predictions, how do you actually practically look and treat data? I know that with the smaller data sets, you try and come up with some kind of rules about how the disease is progressing or who's responding, and then come up with an algorithm or a set of assumptions that then one has to go and test in another patient cohort, or how do you approach this?
Yeah, so the way machine learning works is that you have, let's say, clinical characteristics. You have 100 things, and you know sort of what you're looking for, and so you test which of those 100 things, are there 10 that kind of explain most of the behavior. And then you build that model based on, typically a retrospective analysis of a subset of the trial. So basically, you have 100 patients, you build a model in 70 of them, and then test it in the remaining 30. Ideally, you do that with, much bigger data sets with lots of patients. One of the challenges with machine learning is that you'll often overfit to, meaning that you find characteristics within your training set that explain what's happening in the training set, but then when you try to apply it into a new data set, it doesn't really work that well. I think the scientific literature is littered with examples of that where, gene signatures or whatever just don't reproduce outside of a particular lab, right? So if you test...
So it's an artifact of the data or just some other factor that normalizes the data in a slightly different way?
Yeah, it's more just a characteristic of that particular experiment, right? That gets then carried through, and you don't always know. Like for example, one of the problems with this meaningful data at scale is that you really only have the things that you collect, right? You collect, blood levels of albumin or whatever it is. You do that because we know that they're prognostic, so it's not surprising that they would come through, as one of the variables that may be important for prognosis in a particular trial. What we don't know is what else are we missing, and so one of the advantages of doing these imaging things is where it's everything that's in that image. You don't necessarily have to know what it is. So machine learning in some ways is a little bit more directed, whereas, the AI approaches can be kind of, I guess, quote unquote, unbiased. Yeah. They certainly do have biases, but they may be different ones. And so you kind of do both.
Jane, is machine learning or AI affecting things in your world?
It's affecting all of us. It's a bit like we've talked about statistics. Growing up as a biologist, I never thought I'd have to, reintroduce mathematics and statistics into my science beyond some really basic equations. But it's important for everything, and machine learning and artificial intelligence is here to stay. And I think young scientists out there need to consider that as their training. So I'd like to just turn personal for a moment. You're a trained medical doctor.
Yeah.
How has that impacted the way you look at patients and the way you think about data collection?
Well, I think we're realizing how, there's data being generated every single day in clinical care. And, we need to make that data work for the benefit of the next patient, or actually even for that patient, right? And we don't currently have a system that enables that. Most of the data we generate in clinical care is not contributing to our knowledge base in the way that it should. And so now when I think about, what's possible when someday all of this data that's being generated in routine clinical care is helping advance our knowledge in real time so that the next patient's benefiting from the collective of all this other data, think about what could be possible. How much better we're going to do in really understanding for that individual patient what's the best course of action. I think that's an incredibly exciting future.
And David, how did you end up focusing on biomarker and large data analysis in a clinical setting?
Well, I guess it sort of started with, in grad school where I was sort of trying to figure out what I wanted to do. And it sort of led to cancer biology because that's where the models were. What I always remember from grad school is actually looking down a microscope and, looking at these cells just do crazy things. And, how does that cell that does crazy things come from a cell in a person that's doing exactly what it should be doing?
So how can you understand the genetic process?
Yeah. So that was really sort of the early fascination with it, and that kind of led to, okay, how do you detect these things? And the genetics and the epigenetics, which was really what I did in grad school, and that sort of morphed. Actually, was a good segue into what I do here, which is really related to understanding the genetics and the dynamics of what happens with tumors, when they're getting treated. The technology is sort of where the, data at scale comes from, right? So, even when I started, we were doing things at a very sort of one gene, one drug approach, right? Now it's just everything. Yeah.
And for our younger listeners out there, we've talked about machine learning and artificial intelligence and collecting data, and this is going to include wearables and monitors and all kinds of things. What does the field look like 10 years from now, Mark?
Yeah. Well, I think scientists are going to be data scientists, right? They have to be well versed in data science and be able to work with data of many different types. That what's going to be explanatory about biology, working backwards from the human, the ultimate experimental organism, is going to be a multi-dimensional picture. And I think we're going to want our scientists to be real experts in looking at all of these different data types.
I think we're hearing that they're going to have to be computer programmers and biostatisticians and large data.
It's kind of ironic in a way. So I remember, one of the reasons I got interested in going to grad school was that I read an interview, I think in the New York Times, the Wall Street Journal, about the founder of Netscape, who said that they asked him a similar question, saying, where if you had your chance over again, where do you think this is going? He said, for sure, I'd be getting into biology. I listened to that. Unfortunately, if I'd been a computer scientist, I'd be retired by now. But it seems like it's come full circle, and biologists are now saying, make sure you know how to code.
Yeah, no, it's funny. I remember when I was a grad student, my PhD advisor, I remember asking him, I was doing some yeast experiment, and I was like, maybe we should get a statistician to look at this result. And I remember distinctly what he said. He said, if you need a statistician, it isn't real.
Yeah.
That's what he said. And now I think, man, I should not have listened to him. I should have learned statistics.
I don't know. I struggle like, what does a p-value really mean? Right?
No, but it's just become so. The ability to work with data and work with data at levels that humans can't do manually, right? You got to program. This is going to be essential for the future.
So we've been talking about using current technologies, but beyond biomarkers suggests more than that.
Absolutely. And so, I think it's still the case that there's more information to be had from traditional samples like the tumor tissue and the blood, but there's opportunities to go way beyond.
Okay, so what kind of opportunities are they?
Yeah, so we talk about, using, for example, the smartphone, right? And the way people interact with their smartphone. There's reason to believe that the tenor of your voice can indicate something about your neurologic state. The way that you scroll up and down on a web page, or how often you use the delete key. It's probably not surprising that when you're sleepy or you've had too much to drink, that you probably use the delete key a lot more because your typing is so much less accurate.
So we're measuring hangover response as well.
Well, that's part of your clinical picture, right? So,
Yeah, I think that's absolutely true. I think there are a lot of, you're walking around with a pretty incredible device that tracks lots of things about how you walk, where you walk, how quickly you go up the stairs. All of these things could be used in some way, to measure health. There could be very informative other ways to use those things. I guess your glasses, if they become, have devices in them, you could tell, how long it takes your eyes to focus. That could be, pretty important for various disease settings.
yeah, absolutely.
And of course, there's diet and metabolism and all kinds
Oh
of
my
other
goodness.
factors that get laid into this as well.
Yeah, yeah. No, so there's like when we talk about the microbiome as a supplement, right? The microbiome is like all the bacteria that live in your gut.
More cells than us, yeah.
Yeah, or live on your skin. But there's so many other ohms, that we haven't even tapped into, right? I mean, you're referring to something like the expososome, right? Like the kind of things that are around in your environment. So there's still so many other elements of data that we could capture about a patient that tells us, why they're going to respond to a drug or not respond to a drug in a particular way.
So you think we can simply be agnostic to the data we collect it all and let the data analysis direct us?
I think we have to understand the attributes of a particular data type, like what's noise, what's signal. I mean, we still have to get to that point before we routinely capture it on patients. Because, as they say, garbage in, garbage out, right? If you collect the kind of data that's just impossibly noisy, it wasn't worth it, right? Whereas something that you understand the properties of, where you know how reliable it is and when to pay attention and when to ignore it, that's going to be good data to capture.
Yeah, and I think you don't know what's important until you do, right?
Right, everything can be important. It really depends on the lens. And I feel like with the two of you, we're in good hands. I wish you both the best and good luck in your journeys and look forward to hearing and experiencing some of the outcomes. So thank you, Mark, and thank you, David.
Thank you
Thank
very
you.
much.
What I love about the field of biomarkers, even beyond biomarkers, is that it is still unfolding as we collect more data. And the more data we collect, the more questions we can ask of it. And as we do that, we're going to understand more about disease, how to treat disease, how to identify patients, and really truly get to personalized healthcare. So that's a wrap for today. Thanks for listening. Keep telling your science fans about us. Find us on social media, download the podcast from your favorite podcast app, and feel free to rate us on iTunes. And now, until next time for me, it's back to the lab.